Welcome to this lecture on animal biotechnology. Today, we are diving into a fascinating and essential topic, colony and plaque hybridization. Imagine trying to find one specific person in a crowded city of millions, but you have a magical spotlight that only shines on them. In genetic engineering, we face a similar challenge when looking for a specific gene inside a massive DNA library. This technique is exactly that magical spotlight. It allows us to pinpoint the exact bacterial cell or virus carrying our gene of interest. Let us explore how this incredible molecular search engine actually works. Before we get into the technical details, let us build some intuition. Imagine you are looking for a single, specific recipe card hidden inside a massive warehouse filled with millions of blank envelopes. Opening every single envelope one by one to read the contents would take a lifetime and be incredibly frustrating. Instead, what if you had a magical glowing magnet that only stuck to the exact recipe you wanted? You could just toss the magnet into the warehouse, turn off the lights, and look for the glow to find your recipe instantly. In genetic engineering, when we create a library of DNA, we end up with thousands of bacterial colonies or viral plaques. Colony and plaque hybridization is exactly like that glowing magnet, allowing us to instantly spot our target without testing every single one manually. Now that we understand the basic idea, let us look at the formal definitions. Colony and plaque hybridization are fundamental molecular biology techniques used to screen and identify specific DNA sequences within a genomic or cDNA library. But what exactly are we screening? A colony refers to a visible cluster of identical bacteria, usually E. coli, growing on a solid agar plate. On the other hand, a plaque is a clear, circular zone on a cloudy bacterial lawn. This clear zone is caused by a bacteriophage, which is a virus, infecting and lysing the bacterial cells. Whether we are looking at bacterial colonies or viral plaques, the goal remains the same: finding the one that contains our specific gene of interest among thousands of candidates. Understanding what colonies and plaques are brings us to how we actually process them. The first step in the mechanism is transferring the spatial pattern of these colonies or plaques onto a solid matrix. We typically use a nitrocellulose or nylon membrane, gently pressing it onto the master agar plate. Once the cells or phages are transferred to the membrane, we treat them with specific chemicals to break them open and release their DNA. Next, we undergo a process called denaturation, which separates the double-stranded DNA into single strands. Finally, we permanently fix this single-stranded DNA to the membrane using heat or ultraviolet light. This creates a stable, exact replica of the original plate's genetic layout, ready for the next crucial step. With our DNA securely fixed to the membrane, we can now introduce our glowing magnet. In molecular terms, this is a labeled piece of single-stranded DNA or RNA, known as a probe. We introduce this probe to the membrane, and because it is single-stranded, it will naturally seek out and bind, or hybridize, specifically to its complementary target sequence. The label on the probe, which could be radioactive or fluorescent, acts as a beacon. By detecting this signal, we can reveal the exact location of the desired clone. We can then go back to our original master plate, pick that specific colony or plaque, and grow it in large quantities. This elegant mechanism is what makes the entire screening process possible. To make this process completely clear, let us walk through this visual workflow. On the far left, you see the master agar plate containing various bacterial colonies. In step 2, a nitrocellulose membrane is gently placed over these colonies to transfer them. Moving to step 3, chemical treatments lyse the cells and denature the DNA, fixing it firmly to the membrane. In step 4, the radioactive labeled DNA probe is introduced, which seeks out and binds only to the target DNA. Finally, in step 5, autoradiography using an X-ray film captures the radioactive signal as a dark spot. By aligning this film with the original master plate, we can easily identify and isolate the exact colony that contains our gene of interest. Seeing how efficient this workflow is, we must ask why it is so critical to modern science. Without these hybridization techniques, finding a specific cloned gene would be like searching for a needle in a haystack while blindfolded. For example, when scientists want to produce life-saving medicines like recombinant human insulin, they first have to isolate the human insulin gene from a massive library of human DNA. Colony and plaque hybridization allow researchers to rapidly screen hundreds of thousands of clones simultaneously on a few small membranes, saving years of manual labor. This technique is the absolute backbone of genetic engineering. It enables the discovery of disease-causing genes, the creation of genetically modified crops, and the development of targeted gene therapies. Without it, modern biotechnology would simply grind to a halt. Knowing how important this screening is, we face a critical logistical question. How many colonies or plaques do we actually need to screen to find our gene? To figure this out, we use a mathematical model known as the Clark-Carbon equation. Conceptually, this formula balances our desired confidence level against the size of the genetic fragments we are working with. Let us look at the variables. N represents the total number of colonies or plaques you must screen. P represents your desired probability of finding the target gene, usually set to 95 or 99% for high confidence. Finally, F represents the fractional size of the genome contained in a single clone. If you increase your desired probability P, the required number of clones N will increase, meaning you must screen more plates. Similarly, if the insert size is small, the fraction F decreases, which also drives up the total number of clones N you must screen. Now that we have our formula, let us apply it to a real scenario. Suppose you are screening a genomic library of a small bacterium to find a specific enzyme. We are given that the total genome size is 4 million base pairs, and our cloning vector holds an average DNA insert of 4,000 base pairs. We want to be 99% certain that we find our gene of interest. The first step is to find the fraction of the genome present in a single clone, represented by F. We do this by dividing the insert size of 4,000 by the total genome size of 4 million. This gives us a fraction of 0.001. This tells us that each individual bacterial colony contains exactly 0.1% of the entire bacterial genome, defining the capacity of our library. With our fraction calculated, we can now determine the total number of colonies needed. We substitute our probability of 0.99 and our fraction of 0.001 into the Clark-Carbon formula. The numerator becomes the natural logarithm of 0.01, which represents our 1% margin of error. The denominator becomes the natural logarithm of 0.999, representing the probability that a single clone does not contain our gene. By dividing these two logarithmic values, negative 4.605 by negative 0.001, we arrive at our final answer of 4,605. This means you must perform colony hybridization on at least 4,605 individual bacterial colonies to guarantee a 99% chance of finding your specific gene. Let us level up and look at a much larger scale example. Now imagine you are searching for a specific disease-causing gene in a human genomic library using bacteriophage plaques. The human genome is massive, approximately 3 billion base pairs. You are using a specialized viral vector that carries a large insert of 20,000 base pairs, and you want a 95% probability of finding your gene. Just like before, we first calculate the fractional size, F, by dividing the viral insert size by the human genome size. This yields an extremely small fraction of 6.67 times 10 to the power of -6. This tiny number indicates that each viral plaque holds only a microscopic sliver of the total human genetic code, highlighting why mammalian libraries require so much more effort to screen. Now we plug our new values into the Clark-Carbon equation to find the required number of plaques. We use our probability of 0.95 and our calculated fraction. The numerator evaluates the natural log of 0.05, representing the 5% chance of missing the gene entirely. The denominator evaluates the natural log of a number very close to 1, showing that any single plaque is highly unlikely to have the gene. Dividing these values yields approximately 449,130. This means you would need to grow, transfer, and hybridize nearly 500,000 viral plaques to be 95% confident. This massive scale is exactly why high-throughput hybridization techniques were invented in the first place. We have covered a lot of ground today, so let us summarize the most important ideas. First, colony and plaque hybridization are essential screening tools that act like a molecular search engine for genetic libraries. By transferring clones to a membrane and using a labeled complementary probe, we can pinpoint the exact location of a desired gene among thousands of candidates. Second, we learned that the Clark-Carbon formula helps us mathematically determine exactly how many clones we need to screen to guarantee success, balancing our desired confidence against the library size. Mastering this technique is the first crucial step in isolating genes for cloning, sequencing, and creating genetically modified organisms. You now have a solid understanding of how we find specific genes in biotechnology.